CD40 and CD40 ligand. Do we have
or toll-like receptors?
CD8086 and CD2?
No, it's 40-40.
Or you just learned the 40-40 ligand? Okay.
40-40, CD's 40-40 ligand.
Yeah, okay. All right, so that's the interaction of the B cell and the T cell receptor, and CD4 with the MHC class 2 and so on, okay? Yeah.
I just want to make sure that second circle in the B cell is the MHC, right? That one.
Which one?
That one.
This one here contains it contains the MHC class 2 molecule. Because when you make a membrane protein, any cell has to make membrane proteins, right? And they get made, and then they get shuttled into the Golgi, which has these little vesicles that bud off from the Golgi, right? So that's what that is. It's already made. It's gone through the endoplasmic reticulum into the Golgi. It's bud off, and now the little endosome, the little bleb, is going to merge with this one here. So I don't have that drawn very well, but it'll merge together like that. And then, okay, so those are all bits and pieces of that antigen. And then one of those pieces of antigen is going to go into that peptide binding group of the MHC class 2 and go to the membrane there and be expressed on the membrane. And you have some really good pictures of that process in your book. And the same thing would happen if I changed this from a B cell to a dendritic cell, and I made the dendritic cell phagocytize the antigen. Right? So it's phagocytizing the antigen. The same thing happens, right?
Right.
Okay.
So it's this
all right, so that's the the MHC class 1. How about class 2? How about MHC class 1? So all nucleated cells express it. Why?
Natural killer cells don't get them.
So natural killer cells don't get them. Okay, that's true too. Yeah. So having that MHC class 1 protects it from natural killer cell. But what kind of MHCs are molecules that express antigens. So what kind of antigen is going to be expressed by this nucleated cell that expresses MHC class 1?
What kind of antigen?
CD8.
CD8.
Cytotoxic.
No,
Recognizes.
what kind of antigen?
Oh,
Where does
antigen.
that antigen come from?
From the outside?
From
No, it would be made inside the cell.
Outside.
The MHC class 1, the antigens that it expresses.
me too, I'm dying. I can't even think.
So MHC1 presents antigens made inside cells. What kind of antigens would be made inside cells?
Your own.
Okay, so autologous. So it's going to present autologous antigens, and when it does that, it says, hey, I'm a normal cell, leave me alone. Right? Or if it was infected with a virus, it would, as the virus is replicating, it's making viral proteins. Those viral proteins are going to be expressed by an MHC class 1 molecule to say, hey, I'm a viral infected cell. This is our cells trying to protect themselves or protect our body. I'm a viral infected cell, so kill me. So it makes autologous antigens, it makes viral antigens, or
Tumor.
other, tumor is another one, or other intracellular pathogens. Because there are other intracellular pathogens. There are some bacteria that are intracellular, and fungi that are intracellular. So the cell that is presented to is the CD8 cell, right? MHC class 1 CD8 cells. All right, so if I drew that picture, here's an infected cell, and here's a cytotoxic T lymphocyte. Okay. The infected cell, what do I need to draw on this infected cell? An MHC class 1 molecule. Okay. I drew it differently than the class 2 because class 2 is 2 polypeptide chains and the class 1 is 1. Okay. And here, what's that red thing? If this is our measles virus case, what is that red thing?
Measles virus.
No, measles, an antigen of measles virus. Okay, so it's being shown on the class 1 molecule of the infected cell. And here's our cytotoxic T lymphocyte. What do I need to draw on this T cell that's interacting with that?
TCR.
A TCR, okay. So I'm going to draw a TCR, the alpha chain and the beta chain of the TCR. And then what else do I need to draw in there that's holding on to the class 1?
CD8.
A CD8 molecule, right? Okay. And then we also have co-stimulatory molecules, whatever they might be.
Where'd you draw the co-stimulatory molecule?
So co-stimulatory molecules?
Oh, I got it.
Yeah. Okay. Now, with that information, you should be able to move forward in your packet.
Starting to make sense now. So is this, the CD8, is this after like the antibodies started?
The CD8s,
Yeah,
when do they get activated?
yeah.
Probably about the same time. I mean, you
Oh, yeah,
have...
he did say everything happened about the same time.
Yeah, yeah.
Because the B cells are just targeting the bacteria cells for destruction. Or
Yeah,
control.
B cells are targeting what,
And the
bacteria or
CD8s
virus or whatever.
Are